Hi, my name is Tim Sharipov, and over the summer for the K Fellowship, I research and explore the complexities of the soil microbiome and the effects of Korean natural farming KNF treated soil on the growth and nutrition of lettuce. First, I would like to say thank you to Papa Jack for giving us this wonderful opportunity to engage in independent research over the summer through the K Fellowship. So the intensive use of synthetic chemical fertilizers and pesticides in current agricultural practices creates a range of environmental problems that include groundwater contamination, soil quality degradation, and biodiversity reduction. A different approach to healthy and productive produce growth can actually come from soil microorganisms. The use of fungi and bacteria as inoculants for enhancing crop production is a viable alternative to traditional fertilizers, as they are an important part of the soil ecosystem and play an important role in regulating soil fertility, nutrient cycling, and enhancing plant productivity without causing environmental pollution. So there are many divisions of a plant's microbiome, but beneficial microorganisms are bacterial groups known as plant growth promoting rhizobacteria, or PGPR, that are capable of colonizing plant rhizospheres or the soil surrounding the roots. PGPR promotes plant growth by improving the uptake of macronutrients such as nitrogen, potassium, and phosphorus, and micronutrients like chlorine, iron, and zinc. Nitrogen-fixing and free-living rhizobacteria such as Rhizobium, Pseudomonas, and Bacillus have been found to have positive impacts on crops. This is because atmospheric N-fixing bacteria can establish a symbiotic relationship with the plant, converting nitrogen into ammonia, which is used as a source of nitrogen by the plant. Korean natural farming, or KNF, has been practiced by many farmers, which involves culturing indigenous microorganisms, or IMOs, and introducing them into soil. These types of farming systems incorporate little to no reliance on the use of inorganic fertilizers, but rather advocate for naturally occurring indigenous microorganisms that enhance soil fertility, which should in turn increase plant growth. The subject of my project is the pharmacy plant boxes created by Mountain View Farms in Waianae, Oahu. They are advertised as a combination box of soil irrigation and aeration to foster the natural microorganisms in the soil that are the foundation of natural farming. These microorganisms in the soil, paired with the absence of any chemicals or fertilizers, can lead to nutrient-dense and enzyme-rich produce. Therefore, for my research, I want to see if greater bacterial diversity in the soil microbiome specifically affects crop growth and nutrition. I hypothesize that the diverse microbiome of the pharmacy plant box will lead to higher growth rates and more nutritious lettuce compared to soils with less or no bacterial diversity. My two research goals were to utilize Iolani School's very own pharmacy plant box, as it is what sparked my interest for this project, and to determine the types of bacteria prevalent in Korean natural farming systems. So I collected soil from our very own pharmacy plant box at the One Team Garden. I sterilized half the batch using an autoclave at a heat of 120 degrees Celsius for 20 minutes and planted Iolani cultivated lettuce seeds in seed planters. By sterilizing the soil, I eliminated fungi and bacteria without destroying the soil or drastically altering its chemical composition. So the soil is the same, but there are no benefits of microorganisms. Then 45 seeds were planted in the original soil and 45 in the autoclave soil. I chose lettuce for my experiment because it grows quickly and it's not very demanding. Then, after 2 weeks of growing, I randomly selected 8 seedlings from each soil type and transferred them into separate larger trays. Heights were taken of each seedling every 2 days for another month. An average height and growth rate for each soil was also calculated. Throughout my experiment, plants were grown in an incubator at 20 degrees Celsius with a grow light for half the day, which imitates day and night cycles. I took a total of 4 soil samples during my experiment. 1 sample from each soil type taken at week 0 from before I began planting, and 1 sample from each soil type from 3 weeks after planting. Samples were freezed, and then DNA was extracted using a DNeasy Power Soil Kit. And then a 16S polymerase chain reaction was formed for both samples using a 16S barcoding kit. The PCR product was cleaned and sequenced using a nanopore MinION to identify the bacteria in the samples. After we sequenced the autoclaved and non-autoclaved soils, I found, as expected, that the diversity was greater in the non-autoclaved soil, addressing the first part of my research question. For this relative abundance plot, the more colors you see, the more diverse the sample is. The number of genera is much greater in the non-autoclaved soils, and this was further proven by finding Simpson diversity indices for each sample. The two least diverse samples are the autoclave ones. The sample at 0 weeks had an index of 0.83, and the sample at 3 weeks had an index of 0.97. The non-autoclave soils have a greater diversity values of 0.98 at 0 weeks and 0.99 at 3 weeks. This falls in line with what I expected. However, the autoclave soil at week 0 wasn't fully sterile, as we found the preponderance of many genus of Bacillus. By performing a principal coordinate analysis with the species data from each sample, we can actually see the composition of the autoclave soil is extremely similar regardless of time. This is because it maintains a stable community. For both week 0 and week 3, there is very little change in bacterial composition, shown through the close proximity of both non-autoclave points. However, what is very interesting is that the composition of the sterilized soils changes with time, and that even though the week 3 autoclave soil and the week 3 non-autoclave soil were both very diverse, they were not similar in the types of bacteria. If that top pink dot was down by the blue dots, then that would mean that the autoclave soil becomes more similar to the non-autoclave soil, but it's not. This means that the autoclave soil is becoming more diverse in a different way, and now we can ask ourselves if this difference could affect the growth rate. My null hypothesis was that there is no statistically significant difference between the growth rates of the autoclaved and non-autoclaved soils. I would reject my null hypothesis because after conducting a Wilcoxon rank sum on the rates from week 2 and the beginning of week 3, we found that the autoclaved soil had a significantly greater growth rate than the non-autoclaved soil, with a p-value that is less than 0.05. Overall, my data goes against my original hypothesis that there would be a greater growth rate of non-autoclave soils because of its greater bacterial diversity. As we determined earlier, the autoclave soil was changing between week 0 and week 3. When we sampled the soil a second time, 3 weeks after planting, we found that the autoclave soil is very different. We sampled following this massive spike in growth rate, showing us that the change in the bacterial community composition impacts the growth rate in a good way. As the plants continued to grow, the growth rates became more similar and evened out. This could mean that the bacterial compositions between both soils started to become more similar later on, but I cannot be fully sure. We don't know the mechanism, but through my observations, the plants grown in the non-autoclave soil with more bacterial diversity appeared to be more nutritious. Those plants demonstrated greater amounts of purple and red pigments than the lettuce grown in the autoclave soil. The purple color comes from anthocyanins, which is a vitamin that has anti-cancer, antioxidant, anti-diabetic, anti-inflammatory, anti-obesity, and antiviral properties. Because the diversity of the bacteria is different in the autoclave and non-autoclave soils, there is no purple pigment or anthocyanin within the lettuce grown in the autoclave soil, as some of the bacteria is not prevalent. In my research, I looked at 3 important nitrogen-fixing bacteria, as I wanted to determine the types of bacteria prevalent in a Korean natural farming soil system. The family Rhizobiaceae contains the genus Rhizobium, which forms a symbiotic nitrogen-fixing association with the roots of plants. It was found to be in the non-autoclave soils, potentially influencing greater nutrition rates. Overall, Rhizobiaceae is a small percentage of all bacteria in the samples, and its small amount may not have fully maximized its benefits, potentially resulting in lesser growth rates. The family Pseudomonaceae contains the genus Pseudomonas, which increases the absorption of nutrients like nitrogen and phosphorus. It was found to be in the non-autoclave soils, potentially influencing greater nutrition rates. Like the Rhizobiaceae, Pseudomonaceae is a small percentage of all bacteria in the samples, and its small amount may not have fully maximized the benefits, potentially resulting in lesser growth rates. Finally, the family Bacilliaceae contains the genus Bacillus, which is a gram-positive bacteria. This means that it has thicker cell walls compared to the other bacterias. And in addition, Bacillus species can form long-lived stress-tolerant endospores, which are extremely resilient to harsh environments and high heats. And this is actually why many Bacillus strains remained in the sterile soil after autoclaving. In addition, the fact that it makes up almost 75% of the initial autoclave soil directly boosts plant growth. Overall, my hypothesis was not fully supported by my data because the autoclave soil was a better growth environment for the 8 plants, but there were indications that the nutrient composition was higher in the KNF soil. Potential reasons my experiment did not follow other literature is because my experiment had a much bigger because other experiments had a much bigger sample size, were field grown and not lab grown, and used direct inoculation of bacterial strains. My sample size was 8 plants per sample, but 20 to 50 would be a better estimate of growth. Our results can be explained because soil that is sterilized has been proven by past research to remove harmful pathogenic microorganisms from soil. The microorganisms can leach nutrients from the plant, reducing the efficiency of nutrient uptake, and further stunt plant growth. Because the autoclave soil was partially sterile and only started with the bacteria from the Bacilliaceae family, it has less microbial diversity. Therefore, we can also conclude that it's not greater microbial diversity, but a greater abundance of growth-promoting bacteria, such as Bacillus, that is more beneficial for plants. Overall, a key takeaway is that adding specific plant growth-promoting microbes into soil instead of starting off with a diverse soil is more beneficial to plants. At a population growth rate of 0.8% per year, Hawaii continues to maintain its dependency on imported food and to feed residents and visitors. For Hawaii to move toward self-sufficiency, reliance on imported food and agricultural inputs must be reduced. Hawaii's farmers have tried time and time again to provide locally grown produce, but we have limited space and farmers. Therefore, it is important that we make the best produce possible. The implications of this research for Hawaii is to implement more KNF farming methods to make our state farms and agriculture more self-sufficient and efficient. I am so appreciative of this opportunity I had to be part of the K Fellowship program. I learned a lot during my research process, such as new laboratory techniques, and became skilled with working with DNA. My advice for future K Fellows would be to stay on top of your research, and this will come easier if you research something that you are truly passionate about and will enjoy spending hours on. Lastly, I would like to say some thank yous. First, to Dr. Chen for her time, mentorship, and constant support throughout my experiment. To Ms. Kobayashi for teaching me lab etiquette and supporting me with my DNA samples. And of course, Mr. Hill for helping me with all my data and educating me in bioinformatics. Also, a thank you to Dr. Johanna Anton at Hawaii Preparatory Academy for the opportunity to attend a sustainable soil conference in Waikoloa on the Big Island. Finally, a thank you to Ms. Sakuda as well for her guidance and inspiration for my research. And of course, a big thank you for all of you for coming. Thank you.